Уважаемые коллеги, просьба.
Dear colleagues, could you please take your seats? We are going to resume our work in about 2 minutes. And this will be section 1.
Здравствуйте. Ребята, это ваши. Он будет по видеосвязи, но пока с ним контакта нет, поэтому я буду ориентироваться по ходу.
То есть, а потом будет комиссар?
Комиссаров в записи идет, вроде бы сказал. То есть он записал лекцию, она должна быть у вас, у строителей. Она есть? Можете проверить?
У комиссаров, у строителей, да.
Ну, здесь какие-то есть организаторы. Уважаемые коллеги, позвольте мне открыть первую секцию.
Dear friends, let me begin our first section. It is devoted to molecular epidemiology of SARS-CoV-2. Unfortunately, Mr. Lyoznov, due to administrative reasons, cannot be co-moderating the session, so it will be only me. We have a very full program, and during this section, in addition to all the reports, we also are going to have a round table with our colleagues from Moscow. It will be a video conference connection. So probably we'll do it in the middle of the session. So there will be a short break. Then we'll organize the connection. And then Mr. Kozlov, corresponding member of the Russian Academy of Sciences, will give his report. Right, of course, COVID infection and these 2 years that we have lived with it, okay, they demonstrated not only the relevance, as we heard during the plenary session, the relevance of various molecular, genetic, and epidemiological studies, but also we need to work with co-factors of this infection. Definitely lots of co-mobility, especially when these patients are hospitalized. Okay, the flora and the condition of the flora, and it is usually multi-resistant. Okay, it worsens the treatment, and also there's an outstanding issue of various meta-organisms that may influence the process, which is called COVID infection. So during this section, we will try to discuss all these aspects, and at the end of the session, if we have time, we may have a discussion. So we'll try to answer some questions and maybe identify next steps that will enable not only timely diagnostics of coronavirus, and also all those cofactors and processes, and especially those ones that stay in the post-COVID period. Okay, because the epidemiological assessment related to seasonality of the coronavirus and all its mutations and variability. So this will lead to the situation when this response to treatment is not stable. So the clinicians right now, they're using some metadata in terms of the bacteria, but in each region we will have to assess all those cofactors in order to have success in curing severe patients who are on lung ventilators or in intensive care units. Sorry, I don't have any contact with Mr. Bazikin. Okay, so I'm requesting our technical specialist. Can you please tell me what about Andrei Komissarov's video, because it was sent beforehand. So can we show it? And then we'll see if our next speakers are here. Sorry, I can't hear. Is there someone from the technical committee? Is there Andrei Komissarov's video? Colleagues, I was told that you have this recorded report and you need to just show it. Yes, it is on now. Dear colleagues, thank you very much for participation in this. I'm going to speak about the evolution of SARS-CoV-2. Okay, if we speak about some typical development, the virus load increases from -1st week to the level of symptomatic disease, and then in 3, 4, really 5 weeks, it goes down to the undetected level. However, in literature, there's a number of cases described where we have a longer period of this disease development. These are different publications where such cases are described, and the period might be from 80 to over 300 days. All these cases have something in common. Usually, these are in immunocompromised patients. And you see that the majority of the described cases, this is Hodgkin's lymphoma patients and white cells anemia and other conditions where the immune response is compromised. In August 2020, at the end of August, within the routine genetic oversight over SARS-CoV-2, we identified an unusual genome. It is this 3820. So, and there we saw 6 different mutations in different parts of this genome. In summer and spring of 2020, for the majority of viruses, there was only 1, 2 a maximum 3 changes of these spikes, but here 6 or 7 mutations. Yeah, this definitely attracted our attention. What is interesting, okay, you see that the quoted changes are quite interesting. Del 9670. Why is that? Because this was identified in genetic cluster 5 from minks and people working in fur farms. There was a number of outbreaks in animal farms in Denmark and the Netherlands, and there was a large share of these farms affected, and that is why a big number of minks were removed. And this coincided with those spikes, mutations in the patient I mentioned. When we studied this female patient, she was 47. She had Hodgkin's lymphoma, grade 4. And she hasn't been abroad. She never visited animal farms and never contacted any workers of such farms. And we decided to work it out and find out what was the reason. And she was infected in April 2022. And since April, this patient had had 2 pneumonia cases, and it was confirmed with a CT and clinically confirmed. There were many incidents of PCR positive results. She received therapy for her main condition, and it was repeated rituximab therapy. And we studied samples from the expected source of infection and her samples in 6 periods. The major question we had whether we are dealing with some reinfection or is it a persistent virus. Then sequencing results and the genetic analysis that was carried out, it turned out that this virus in patient S, it has one clade and it is one line, and reliably we can say that it is not a relative of cluster 5 viruses. And in August 2020, there were 12, 18 mutations in comparison with in January of 2021. From 10 to 21 mutations were added to that. So the overall number of mutations reached 40, and 18 out of 26 viral genes were affected. When we compared the speed of evolution of SARS-CoV-2 in this patient with the average speed of evolution of such variants as B17 and the British strain, it was demonstrated that the frequency of changes is about 15 to the power of 10 minus 4. And this is worth mentioning that such mutations were not related to COVID plasma prescription and application. So, and plasma didn't lead to neutralization of viral elements. And what else is interesting? So these spike protein mutations that was observed in a patient S, but we can see it in other variants as well, alpha, beta, and delta. We see here that these mutations or changes and deletions, they are in similar parts of the spike and that original patient S, she demonstrated the same deletions. Okay, they were appearing, and then, for example, del 6970. This is typical for the British variant B117, and also a number of changes specific for the Delta strain. When the immunology, the immune response was studied of this patient, it was demonstrated that B cells are practically absent. Then immunoglobulin of G class, again, it is absent. Then no neutralizing antibodies to SARS-CoV-2, and T cells were expressed. And you can see it on the right. So, and in gamma interferon and alpha, okay, when T cells are stimulated. We did the full zone sequencing using the commercial asset for NGS. We wanted to identify alleles of patient S. We identified them, and this made it possible to forecast mutations in this genome, and especially in expression of peptides in the virus from this patient. We used the model, and it was possible to forecast 5 changes that might affect these peptides. First of all, 2 changes of non-structural peptides. It is they are all shown on the right, and also 2 changes in the spikes. So you see that according to the calculations, with the help of this model, we see what might theoretically influence this presentation of these changes. Okay, which peptides are presented by T cells? We used the ADB database, and we identified 12 changes in these peptides that we received in our experiment. And all this leads to the reduction of peptide expression. All these changes, they have been strengthened in virus in patient S. Probably the driver evolution in her case was avoiding CD8 T cells. And in order to check all this experimentally, we ordered peptides with mutations in NSP3. And then we manipulated with the cells from patient S. And we demonstrated at different periods that these mutations and mutant peptides with all these changes, all this leads to a reduction of activation, significant reduction of T cell activation. Identified changes. Okay, so they lead to the condition, and this relates to all other alleles. And here, these red dots, they show the difference in terms of the ability to represent peptides in these changes in different HLA sections. The gray color shows some other elements which were not met in patient S. Right. This was true only for some type of molecules. But as for second class of molecules, this was not true. So this variant developed in this patient, okay, it could avoid this T-cell response in other patients as well. And their HLA alleles were different. So the conclusions. So we have described the longest case of COVID-19, and it was 318 days. And virus developed up to 40 mutations, and 34 of them stayed within this genome. And the major factor was avoiding T cell activation. And this type of virus didn't spread in the population. At the same time, it should be said that if we deal with immunocompromised patients, in the organisms, the virus might mutate, and those variants might be evolutionarily successful in order to spread in the general population, not immunocompromised people. And this reinstates the importance of studying specifics of SARS-CoV-2 and the importance of protecting immunocompromised patients from getting infected with SARS-CoV-2. And this includes the herd immunity, because it reduces the chances for these people to meet those people who are infected. This work is the result of a number of organizations and people. This is a preprint version, and it is on Reprint Research Square. It is available. And here you see the list of all the people who contributed a lot to these studies, and it is Skolkovo Institute of Science and Technology, especially Yevgenia Alexeyeva and Georgy Bazzikin. Then Institute of Problems of Transfer of Information. Then people from our institute, Institute of Bioinformation Technology, clinicians from the First Medical Institute in St. Petersburg, especially Oksana Stanevich, and people from the City Hospital 31. And my special thanks go to Dmitry Cherdakov from the National Medical Research University, named after Pirogov, for great participation in the immunology part of this work. Thank you for your attention.
No, since this presentation was recorded previously, we cannot ask questions. The first speaker is Georgy Bazzikin, a professor of the Skolkovo Institute of Science and Technology. He will speak about the genome epidemiology of Delta strain in Russia. Good afternoon. Well, thank you for the invitation. It's a great honor for me to be with you. I'm sorry I cannot be with you personally, but I will describe our work associated with the Delta strain and the current epidemic of coronavirus in Russia. It's not a secret at all that coronavirus hit Russia, that it affected Russia severely. And Russia, over the pandemic, has been among the 5 countries with the maximum number of registered cases. But genetically, our epidemic is not that well researched and studied. Here you can see the GDP of different countries. The dots are different countries, and a vertical axis demonstrates the share of the calculated genomes in the GISAID database, which is the main resource for the exchange of genetic information on COVID. And This logarithmic scale demonstrates that there is a certain correlation here. The richer countries present a lot of data. Russia is below the trend. South Africa, with lower GDP, provides a large amount of data, which makes it possible for them to discover the new strains quite early, like Omicron, for example. We hope that the situation is improved because it attracts attention, and this article says about that Russia is among the countries that do not know that well what is happening genetically in the epidemic. So we're talking about the open data, of course, in this case. Now, in order to contribute into the tackling of this issue at the beginning of the pandemic, almost 2 years ago, the Russian Consortium for the Sequencing of Coronavirus Genomes was created, Corgi. A lot of different organizations are part of it. It's called TECH. We're responsible for the data analysis for the evolutionary, first of all. And so when we discussed epidemiology, genetic epidemiology of COVID in Russia half a year ago, there was quite a lot of interesting things. There was a great diversity of lines. We have endemic lines, alpha, for example. But then after that, the situation with the coming of Delta changed. It became very dull and very uniform, and so Delta just ruined the whole diversity. This is typical not just of Russia, but of the rest of the world. Here you can see each country is represented, time and share. The green color is responsible for Delta, and we can see the Delta is everywhere, and Delta ruined everything that existed prior to Delta. There seems to be nothing interesting at first glance, but in reality, Delta by itself changes. It is under evolution, and it acquires new mutations true to the coronavirus of the wild type. And if we build the evolution tree of all the coronaviruses, we will see that within Delta there is quite a serious genetic diversity accumulated, and we are trying to monitor the variants that exist in Russia, following the open data, of course. We said we have the portal there, which allows us to follow the dynamics of different versions. And Delta came to Russia. Well, at the beginning of May, it became seriously spread, and then its frequency increased from about 1% at the beginning of May to almost 90% at the end of May, and so the speed of growth was about 10% per day, which is a bit higher than with the neighbors. In Great Britain, for example, it was about 7%. Possibly this is connected with the fact that in Britain, Delta was spread along with the Alpha variant, Alpha strain, and the Alpha strain restrained Delta in a way, was a competitor in a way. But in Russia, it was spreading along with less adaptive variants, and there was no strain that could restrain it. So it spread faster. But when we began to look at the specifics of the Russian sequences of Delta, we discovered that they carry practically all those two mutations that are rare for the rest of the world. The vertical axis here shows the share of those in the samples, red from Russia, and the blue non-Russian samples. And the first two, both of them are associated with the Mann-Slot's replacements and substitutions, and they have the frequency over 90% among all the Delta cases in Russia, and they're quite rare in the rest of the world. And when we almost at the end of the study, the carriers of the first mutations and the evolution branch were associated with them was named AY122, but in Russia they're interconnected. That's typical of Russia. And the incidence here was quite high during the pandemic, but it was quite high at the time of the Delta's presence in Russia since May. But actually, from the first time it came to Russia. Now, it was interesting for us to find out why a certain particular variant of Delta began to spread in Russia, because the situation was radically different, because we could see the onset of the pandemic and we could see that virus was being brought into Russia, imported for Russia many times. Maybe that was because the passenger flows were not that active and there were not that many strains being imported at the beginning of 2020. The constraints there were severe on travel, not that severe in 2021. Maybe there was certain adaptive advantage associated with that, but there is no real data. These are not the mutations in spike, but for those who are, well, but anyway, 3 hypotheses associated with incidence with different aspects of genetic drift. Maybe there was some sort of an event of super dissemination, super spread in Russia. Maybe there was an effect of the primary case, and because of that first case, it began to spread. There were certain variations there as well. We can exclude the hypothesis of low import. We can see lots of imports of different variants of Delta. There is the global tree, the blue. These are the international samples, red Russian samples. You can see the major import that brought about the spread of AY122, and we can see that there are numerous. This is a smaller tree. Numerous other imports of different Delta variants. And we can see another way of representing the same data. Horizontally, there is a date, and the dots are individual samples taken on particular days. Large dot, many samples. The regions are indicated by color. We can see individual lines. These are the contributions of the Delta associated with the imports. And we can see there are many of those imports, and only one of them was evolutionarily successful, the third line from down below, IY122. So it began to develop, while the others were there for a while, but were never dominant. And why and how can we explain this genetic picture? Russia is unique in this sense, or rather unusual indeed. Here in the upper left corner, we can see the countries where the epidemic of Delta is more uniform, where genetically similar viruses there are demonstrated there. There is a lot of variants of Delta associated with one import. Such countries as Russia, Japan, Australia, where the borders were closed and there was very little import. What is Russia doing in this group of countries? I am not quite sure at all. Although I must say that we were very much concerned with the fact that these mutations provide the virus with certain advantages, but we really do not think so. Mostly on the basis of the dynamics in other countries. Combination of mutations of this type was observed in other countries, in the neighboring countries, in Kazakhstan, in Armenia, Finland, and nowhere this combination of mutations grew as radically as in Russia. Everywhere it was lower or higher, but it was always just parts of percentage. So it was late growth in the summer of this year was still not very high in those countries. So we can exclude the hypothesis that the matter was associated with the lower imports. No, we are not sure at all whether there was any adaptive advantage of this combination of mutations. It doesn't look like that. We do not have any sort of a super event mentioned at all, which could lead to this high percentage. The effect of the first patient, well, in Russia, Delta was imported not so many times, but at the beginning of the epidemic of the Delta, global epidemic of Delta in March and April, where there was not that much Delta in the world, and each and every import could be more or less successful. And just one of the variants bringing of those mutations was successful, and it spread for a very simple reason. Every variant of Delta brought in those days had advantages as compared to the wild virus, and the others, the ones that came later, did not have that much advantage, because by that time we had developed our own variant. So the genetic analysis demonstrated that it was imported to Russia dozens of times. That's what we can see. Of course, there was a lot of imports that were not even taken note of, but there was just one subline that was spreading in Russia. It was, I think, associated with the genetic drift. And there is no data supporting that. And the variability of Delta and of Omicron requires careful surveillance in Russia. And then at the beginning of the global spread of the new variant and new strains such as Omicron, each and every import can be of great significance. This work was done by a large number of people from different organizations. In Skoltech, the key author was Galina Quint. This paper was recently published as a preprint at the site of preprint. We have time for questions. I have a question. Allow me to begin. Now, are you trying to forecast the speed of the spread of Omicron in Russia, considering the data for Delta? No. As for there is not that much Omicron in Russia, and we do not yet have data on the internal transmission. All the cases are imported cases, so we can just rely on the external data for other countries, and we can just say that the epidemiological situation in South Africa is not that much different from that in Russia, and there the wave of Delta has recently was attenuated, and although quite a lot of people there suffered from Delta and have not that many in South Africa, either in South Africa or here, but I'm quite sure that the when we have more cases of Omicron, then we will be able to carry out some analysis of the situation here, because it says we know that the Omicron strain is spreading much faster than the other strains, and we will be able to make assessments only when we can face it here in Russia. Any other questions? Well, thank you. Thank you very much. Thank you for your presentation. Allow me to introduce our next speaker, who is going to be with us in person. That is Elena Ilyina, who will speak about the variability of microbiota of respiratory system with patients.
Thank you very much. First of all, I would like to thank the organizing committee and Anna Yurievna Popova for this invitation to give a talk at this important conference. And I'm not actually going to speak about genomics of COVID as it is, but I am going to speak about variability of microbiota of the upper respiratory ways among patients with severe and light form of COVID-19. John Tony formulated this notion some time ago, and when you speak about microbiota, so we view a human organism as not only various eukaryote organism, but also other organisms as well. When we were reviewing natural biotopes of human beings, so we were taking great care of analyzing skin and gastrointestinal biotopes, but not respiratory ways. So microbes in the upper respiratory ways remained an interest only for pediatricians and also pulmonologists who were speaking about pathogens of various pulmonary conditions, Hobble syndrome, and so on. And in terms of the normal functioning of the upper and lower respiratory ways, there were not many. And in the pandemic, researchers turned to microbiota of respiratory ways, including or specifically the upper ways. When we analyze this microbiota, okay, so there's a nasal cavity with a lot of Staphylococcus, then in the throat where we have Carina bacteria and Maraccellas, and then in addition to cocci, there's also Haemophilus and Diserium. And when we consider this nasopharyngeal test, and we have been doing it for several years already, so we see here, we get here a complex, a set of flora in these two areas, nasal and pharyngeal. And on the basis of analysis of this flora, we are speaking about our attempts to modulate and understand development of a COVID infection on the basis of the analysis of this flora. Right, what did we know from previous research and literature? What is microbiota of the respiratory tract in terms of viral infections development? Microbiota of respiratory tracts, like gastrointestinal microbiota, okay, they take part in the overall immune system, and it takes place everywhere. And the bacteria in the respiratory ways and their metabolites, they may prevent development of viral infections, and they have a protective function. For example, neuraminidase, and so okay, it prevents flu infection from spreading. Then specific bacteria, on the other hand, may promote some disease development. For example, Haemophilus influenza promotes respiratory virus development. It should be noted that we have data on COVID when this potency factor is identified for this virus as well. And secondary bacterial infections, they lead to serious complications and lethal cases if they are comorbid to coronavirus infection. Right, so this bacterial infection as a threat, this was the major focus of research works in the metagenome analysis, and it has been demonstrated that among bacterial pathogens associated with severe development of COVID-19, there's Mycoplasma pneumonia, and then after it, nosocomial infections. And this is aeruginosa, Haemophilus influenza, Klebsiella pneumonia, Helicobacter, and so on and so forth. there haven't been many works like this, and as I said, this has been developed largely due to the pandemic. And the goal of the study that I'm presenting was to study the variability of microbiota of respiratory ways in patients with different levels of disease development. And this research was done by a consortium of a COVID-19 study that was created in April 2020, and it is supported by a grant from the Scientific Foundation. So the main blocks of samples, it was nasopharyngeal samples. They were taken in different cities of Russia, and clinics in Moscow, Nizhny Novgorod, Irkutsk, and Kazan provided this material. Then all these samples, we identified DNA and RNA, and there were two lines of sequencing. First, it has the totality of information on microorganisms in each sample through the variable section, and also information on the full sequencing of SARS-CoV-2 with the help of a primer panel, RTIC. So all data on genetic sequencing is incorporated into GISAID database and the Russian VK-RUS database. So all this information is available there. A couple of words about SARS-CoV-2 genomes. We began collection of these samples during the first wave, and then we completed this during the second wave. So there was no great diversity of strains, mainly genomes of SARS-CoV-2 that were identified among patients included into this study. They were B111 line. And for our purpose, this was good because we understood that we have approximately similar genome landscape, and this will not prevent us from assessing the bacterial microflora into development of each specific case. After analyzing the data that was received for these patients, I'd like to remind you it was 4 cities which are quite far away from each other: Moscow, Nizhny Novgorod, Irkutsk, Kazan. We evaluated the totality of all the metadata. And what can we conclude? So we see that there's approximately the same number of people who were hospitalized and outpatient. And those who were hospitalized, there were those who needed additional oxygen support, maybe those who didn't need it. There were patients with different level of lung damage, and also patients with hypertension or without it. Analysis of the data was done using bioformatting methods. And on the one hand, they were aiming at identifying some stable compositions and searching for associations with the level of severity of disease. Then we added some external data we received from patients from Alicante, Spain. So approximately at the same time they were doing a similar research, and the results were published at the beginning of this year. And various differentiated organisms from gastrointestinal tract among severe patients. Right. What are the conclusions? Firstly, it was possible to confirm and demonstrate that the respiratory tract biota, like GI biota, forms some balanced clusters within which a certain complex of organisms exist, and they have some metabolic ways of interaction. And we see 6 clusters like that in our sample. Probably it's hard to read this slide, but believe me that these 6 slides, their incidence was high in different regions, in those different cities. And those samples that information on which came from Spain, it was also obvious there. So this makes us treat this quite seriously. So this is a realistic picture of various associates of respiratory tract. And this is a distribution of these respiratory types in relation to the severity of the disease. If we speak about different microbiota, we consider different parameters. For example, there's such a parameter as alpha diversity. And there's a Shannon index, which is used. And so the higher it is, so the more diverse it is, the more healthy biota it is, because the immune system is quite high. So there's no unified cluster of bacteria which occupy the whole niche. So there's respiratory types. They have different alpha diversity index. And the 5th one, there this respiratory type index was very low. And in the 1st and the 6th, this alpha diversity rate was the same. So, and we saw patients of the same type with a different level of damage. And we see here that respiratory type of 4 and 5, they had the greatest lung damage. And if we look at oxygen demand, then we see that the greatest alpha diversity, there were lots of patients who didn't require additional oxygen. And in the 5th type, the picture was different. And there was this 6th type, which is quite healthy in terms of the severity of COVID-19. All these patients, they didn't have any serious lung damage and didn't require oxygen support. And I'm speaking only about hospitalized patients. These are not outpatient cohort. And they all had clinical symptoms. And analysis of specific groups of bacteria demonstrated that the more serious cases are associated with Rhotia bacteria. They are dental pathogens, and this was described earlier, the association with COVID-19. And the lighter cases, Leptotrichia bacteria, they produce butyrate, which is a metabolite associated with prevention of coronavirus infection development. So we see these well-balanced communities and respiratory tract biota has a link to the severity of cases, and several respiratory types of biota are associated with more severe cases. And roti, lactotrachea is less severe, and rotia more severe. And a couple of words about future research. Definitely, research in the sphere of microbiota studies in association to the severity of diseases. This is a very promising area, and this might enable us in the future to forecast how not only COVID-19 but other serious infections might develop. Flu, for example, judging on the respiratory biota of patients. Thank you very much. Thank you. And here you see the team of researchers. Okay, and this is done by the academic community consortium. Thank you very much. The time frame. Yeah, so we are short of time. So next speaker now.
Allow me to announce the first speaker for the roundtable discussion. So that's going to be Roman Kozlov, corresponding member of the Academy of Science. He will speak about the antibiotic resistance at the time of the pandemic. Good afternoon, dear colleagues. First of all, I would like to start by expressing my gratitude to Madam Popova for the invitation to take part in this important forum. My task is quite difficult. I'm planning to describe what is happening today with the resistant microorganism and what has changed in the attitude and with them, and what we're planning to do in order to minimize the risks of the spread of resistant microorganism. Well, it's not my place here in this audience to speak about the discovery of antibiotics. This unique class of drugs, I was going to say this, here is the son of Alexander Fleming, who studied the colonies of Staphylococcus, and it's not surprising at all that there is a mushroom here which is called Penicillium tatum over there. You can see it to the right. And Fleming, when coming back to work, discovered that there, next to it, there was Staphylococci colonies, leases of those, and obviously that particular fungus produced some sort of a substance that worked against Staphylococci. But the real experience of the application of antibiotics goes back to 1940s, and we should remember the name of Zinaida Yermolieva, who created the Soviet penicillin using penicillin crustosa, very different fungus, penicillin crustazine. She also founded the Soviet Institute of Antibiotics, and in those days the Soviet Union had a priority in the full cycle of the creation of antibiotics, and many of the drugs were developed there and were manufactured in the Soviet Union. Allow me to remind you of what antibiotics have done. Just look at just one number. This is a hospital in the middle of the 20th century, and there was no proper infections control from the point of view of the clinical pharmacology. The drugs were used in the wrong way. Penicillin was used twice a day. But what has it done? Indeed, in those days, the mortality went from 9 among 10 to 1 among 10. And the chance for the pregnant mother to die was 25%, every 20%, and every fifth woman died. And penicillin, the first antibiotic that was wrongly administered, was a fantastic achievement in this field. In the Russian Federation, over 200 antimicrobe drugs are registered. Antibacterial are represented in 20 classes. Antivirus drugs are actively being developed, and many noticed invasive mycosis, and these particular drugs are also being developed, and they provide for the safety of our patients. Antivirus drugs, first of all for HIV and hepatitis, and protozoa, mostly for the malaria treatment. If we look on the whole at the prospects and development of new antibiotics, I should mention one thing here. Founding thing, indeed. We should understand that resistance to antibiotics and antimicrobial drugs is an inevitable fact. When we read in literature focused on the mass reader that there is a new drug discovered in weeds, and there is no resistance to that, we should be very careful about this publication. Resistance is inevitable, and the goal of all the medical professionals and non-medical, we are talking about the concept of health, is to minimize the appearance and the spread of resistant microorganisms. Here you can see the story of some of the antibiotics since their discovery and up to the emergence of resistant strains. For some, it was just several months, for some years, but it's an inevitable biological phenomenon. So what should we understand, dear colleagues, under the present-day conditions? Antibiotics and the resistance to those affects our struggle against infectious diseases, cesarean, target therapy in oncology, transplant of different organs, and from the so this should lead to the 4% growth of VVP in the world. If with sustainability or resistance we do not fight, then unfortunately by 2050 the death among the new mothers might increase 50 times. And so what we're talking about is important not only in the context of infectious diseases, but also for the whole system of the public health and protection of health in general. The major problem on the whole is the lack of knowledge. We're talking about professional publications on the one hand, but on the other hand, we should understand well enough. And now we're most grateful to the government of the Russian Federation and our friends and colleagues from Rospotrebnadzor who worked on the strategy of the prevention of antimicrobial resistance in Russia up to the period of 2030. It's very important to inform the population about that, and this is a priority. What we had prior to the pandemic, there was quite a lot of surveys of the public opinion. So are the antibiotics killing viruses? The answer seems to be obvious, but look at the statistics. Yes, 45% said in 2011, and the latest data available, apart from the Znania Society where we spoke, the data for 2019 and 18, and I think they're promising because they demonstrate that the number of these people, although is still quite high, but it is lower. They're optimistic because if the information campaigns are properly organized, and if these campaigns are actively supported, and if they are continuous, then the impact on our population will be effective. One of the programs we're carrying out is in Smolensk region, and now it is scaled as a pilot project in 4 cities. And I hope very much that by the end of this year, beginning of the next year, we will have certain data to assess the real impact of these programs in the bigger cities. Another problem I would like to draw your attention to is evolution, although, of course, it's something very trivial for the professionals. The evolution of viruses, Staphylococcus, Streptococci. Now, in the 1960s, there were developed drugs against gram-plus microorganisms and then gram-minus bacteria. And about 70% of all the infections were associated with those, and the evolution arose from monoresistance to polyresistance to pan-resistance, and there is resistance to practically all the antimicrobial drugs we have. Why do we need to speak about that? Because our understanding of how significant the problem of resistance is became obvious for the global community. In our country, they began to understand this much earlier. But in 2014, Jim O'Neill published a report concerning that. He said if nothing is being done, and this number has been mentioned, then the summary loss of GDP is going to be about 100 trillion rubles, and 250 trillion by 2050. That's enormous. And then another number that is very important and has an impact on the necessity to make joint decisions. And you can see it on the slide. We can see not a single country in the world, not a single continent, irrespective of the gender specifics, ethnic specific features and geographies, free of the problem. It's the worst scenario that is forecasted, and we are quite sure that we are not going to reach that level, but it shows how significant the problem is. And another number that is impressive and used by journalists quite often, if today, according to the experts in the world, there are about 700,000 people dying of infections caused by resistant microorganisms, if we don't do anything at all, by 2050 it's going to be 10 million people, which is more than the number of people dying from oncology. There's 2.8 million. So, well, according to the experts from Respiratory, in Russia, there are about 2 million deaths associated with medical infections. And many of the strains are resistant to several classes of antimicrobial substances, and about 64% are resistant to one antibiotic. What does it lead to? If there is no proper microbiological laboratory with the absence of data on sensitivity, there is practically no chance to have the proper therapy for the patients. It's less than 30%, and I'm most grateful to Anna Yurina for the support of the professional standard on medical microbiology. For us, it is critically important. We do need professionals who would have the competences of bacteriologists, parasitologists, mycologists, and because it's a very important profession, and we're waiting for the Ministry of Education and Science a special deed pertaining to that. We need this standard. Why are we talking about that? Because we should be very careful about antibiotics. On the one hand, you should understand what is happening. There's a segment of the use of antibiotics prior to the pandemic. Our country always looked not that bad, not that poorly. If we look at the we were at the level of the best practices of Western Europe, at the level of the Netherlands, Germany, Sweden, and other countries with low level of the use of antibiotics, many times lower than Eastern Europe. Historically, they use a lot of antibiotics, and there is no need to remind you of that. The high is the use of antibiotics, the high is the antimicrobial resistance, and so we can see what is happening in Russia with the systemic antibiotics in Russia. But through education, we could modify the system. What happened with COVID? Colleagues, the use of antibiotics in hospitals increased by 28%. 4.1 DDD per 1,000 people per day. And what is even worse, and which inevitably will lead to the growth of antimicrobial resistance because of the two drugs, because of zithromycin and levofloxacin, because those drugs, which according to UOH is a reserve group of drugs. Now, everything is interconnected. We can see the examples of poor the use of countries, of antibiotics in the countries. In the United States, the antibiotics are not banned as growth stimulators in cattle breeding, for example, or in poultry growing. And look at the use of antibiotics in agriculture is 10 times higher in the United States in agriculture than for humans. And again, there is no need to speak about the circulation of antibiotics in the environment, in veterinary, in agriculture, and all these studies we're carrying out within the framework of the health programs should be, first of all, focused on our patients in clinical practice. A very special problem. Antimicrobial resistance is associated, of course, with the quality of drugs. Our data indicates that not all the drugs that we have at the pharmaceutical markets of the Eurasian unity are proper quality. There's a pseudogenetics. There's a lot of insoluble admixtures in those, a low level, lower than declared level of the concentration of the active agent and fast growth of resistance. And the group of Professor Zurianov has similar data, and our data with our colleagues from Ukraine demonstrates that among of the declared 500 milligrams in some of the pseudogenetics, there was just 375. The raw level of macrolides in case of coronavirus infection is a direct way to antimicrobial resistance. We spoke a lot about the vaccines, but here is what is happening with antibiotics. Antibiotics for pharmaceutical companies are not that profitable. Look at the 18 companies that used to work on the antibiotics. By the beginning of this century, only 5 of them survived, which is an absurd situation, but this is true. One drug in this development requires 7 years and $1 billion, and for pharma, now, it's easier to work with antidepressants. The patient is going to keep taking them all through life, which is very different for antibiotics. Antibiotics are short-term drugs. And so the system of registration is, the recoupment period for the development of antibiotics at present is 8 years. And so it is very important now to make sure that more companies work on antibiotics for the market. If we look at the antibiotics, the golden age for the antibiotics came to its end in the 1960s, because those were obvious discoveries, the low-hanging fruit, so to speak. And then the number of the new drugs went down. And the role of the state is very important in these fields. Look at the trends. Thanks to incentives, here is what we had before the incentives appeared. Thanks to the incentives provided by the governments, the number of drugs for clinical practice increases. We are following the new approaches that are being planned by 2 countries, Great Britain and Sweden. The so-called assistance to the farm companies for the acquisition of drugs by subscription. When the state pays a certain amount of money to the pharma company, the company is not advertising the drugs, and the government fixes the price and decides where to buy and how to use the drug. In theory, this looks beautiful indeed. We are following these approaches, and in the United States there is also the Pasteur law that works in a similar way, but there is a system of public health that is somewhat different. Our colleagues from Sweden are doing something similar. But historically in Sweden, they do not use that many antibiotics. It's very important for us to know how the models work. Another important thing, almost at the end of my presentation, is associated with the technology, with evidence based results. I'm talking about vaccination. Vaccination with bacterial and antiviral drugs, because the lower use of antibiotics is better for all of us, and there is no need to remind you of the new vaccines and how they are being developed. The 21st century is the age of the platform decisions for vaccines, and we can fortunately see many of those. What needs to be done in the future? Now, the national policy of the application of antimicrobial drugs following the regional local specifics of resistance is an old slide. The interest to the antibiotics has grown, which is very good indeed. There is the resolution of the government number 2045 on antimicrobial resistance, and we can show quite a lot. In the field of the awareness program, professional competencies, the creation of new diagnostic systems, the monitoring systems. I'm talking about the map of antimicrobial resistance, the certain constraints for the use of antimicrobial drugs, and what is very important, and I'm happy to mention this at the conference, something classical, the international and interdepartmental cooperation. We're most grateful to our colleagues from Russian Academy, from Rospotrebnadzor, for the joint projects we're carrying out, because all these are extremely important. I also would like to mention at the end of my presentation the following. At present, with the new information technologies, we're going to work with those. But finally, just one thing to mention. For us to understand very well what is happening, there will be no effective antibiotics and effective treatment without antibiotics. Antibiotics should be loved and carefully treated. Thank you.
Right, then let's begin this roundtable discussion, and I hope our colleagues have connected. So I'd like to give the floor to Mr. Kulabykhov and Trotsenko. I'm sure we all know them very well. They work in the most difficult conditions. Okay, they organize help for people with COVID-19, and they organize intensive care. And they are on the front line of this fight for the life of our patients. Hello, yeah, we are connected. Hello, yeah, we see. Denis Nikolaevich, let's begin with you. We are very much interested because we have just listened to a report that a lot of antibiotics are taken. So what about hospitalized patients? What do you see? What about microorganisms? What about complications? What is happening now? And what do you expect in terms of healthcare system and epidemiology? Okay, thank you very much for this invitation to take part in this roundtable. This is an important question. And first, we should say that the general population is not well informed, and our colleagues in Germany, for example, okay, so they have programs like surviving sepsis. Okay, they tell just lay people about hospital infections, about resistance. It exists in Germany, for example. We see that our population does not fully understand, and lots of people believe that patients with the coronavirus infection, and actually we managed to deal with cytokine storm and respiratory support. Methodology has been expanded, and all extracorporeal oxygenation methods. So we now know how to deal with this. At the same time, bacterial superinfections, this is something that we still cannot cope with. And patients with COVID-19, they need all those biologically active and interleukin blockers and so on. And the therapy they receive together with immunosuppression and additional immunosuppressive therapy. So there are all prerequisites for bacterial superinfections. I cannot say that now we see certain changes of microbiological landscape in comparison with the period of pre-COVID times. So we know what we are speaking about. But what we see now is that definitely this resistance is growing. And if 10 years ago, for example, for intensive care unit doctors, carbapenem plus rinizolit, this was a joker card in our pocket, and it was possible to cover the majority of pathogens, and then we could be sure for 72 hours. But now microbiological monitoring that we are doing within our clinic, and we use online platforms of Smolensk colleagues as well. So we see that now we don't have this joker card or trump card. This resistance is so diverse. And basic pathogens like Klebsiella Helicobacter, Escherichia coli, and they are sensitive to carbapenem only in 55% of cases. So this empirical therapy, you never know. It's like you toss a coin, and it is 50-50 that it will work. So local microbiological monitoring, not only within one big hospital, but in each intensive care unit or department. And we, in our Kommunar hospital, we have 13 of them. So this is the work which is being done all the time. And this training program, so we teach how to stratify risks of infections of these antibiotics resistant strains. So we organize all this, introduction and development of new protocols. And what is interesting here, in May, at the annual conference, we are going to talk about these results. So patients with severe development, they are now in ICUs. We take a sample from the thick intestine for microbiota analysis, and very interesting results demonstrate that many patients who come to us, not from other hospitals, but from home, and Escherichia coli, so they have it, and it is pan-resistant. So partly, this is the price of antimicrobial therapy and drugs for outpatient cases. So this is the most relevant question for all medical specialists. What is worse, and what might kill the humanity? Microbes which are resistant to drugs, antibiotics, or viruses? This is an outstanding question. Thank you very much. Vladimir Vitalyevich, you also have very rich experience in your multi-profile clinic. So what do you see? What your intensive care unit specialists, what do they see? So what about circulating viruses, and what is going to happen with that? Good afternoon, dear colleagues. Well, I fully agree with the previous speaker. Yes, this is an enormous problem, and all of this, appeared not yesterday. Now this COVID-19 highlighted all these problems. And this is not only typical for Russia. And the United States, for example, 72% of patients with COVID, they received antibiotics, and you know that only 10% they have bacterial pneumonia. And out of people with artificial ventilation of lungs, they are in a risk group, and they often develop bacterial pneumonia, and the cause of death is this type of pneumonia. And you see the dynamics. Yes, it is growing and expanding, and probably in the near future this won't change. Maybe we'll see that different organisms will be shown. So we saw today in one of the talks that so a certain bacteria is the most prevalent, and we have Klebsiella Helicobacter, and yes, they are pan-resistant, or at least some of the strains of them. And of course, it is extremely difficult to treat these patients. What do we see? Okay, lots of resistant strains due to wide use of antibiotics, then the lack of new antibiotics. And a third problem highlighted by coronavirus, this is diagnostics. Although we see that great efforts, new laboratories, and the new specialty of this microbiology, it was established, but we need a lot of new microbiologists, okay, and some new equipment won't help or won't make it possible to diagnose immediately all this. So we need to develop this, and we need new training programs. And I believe in the past 2 years, the level of intensive care specialists went down because all these interns, okay, they had to work in COVID hospitals. And this should be improved and changed. Okay, the training system should be changed, and microbiology, there should be more courses. We don't have much time, and I believe the problem we speak today is when the hospital infections now and the strains, the outside hospitals, and we are facing this directly. We see these patients with resistance to these specific strains. So all these problems, they are really serious, and we need investments in this. There should be consortia, research consortia, and clinical institutions should be part of this. And such consortia will help us to deal with this in a timely fashion. There's a gap between fundamental science and clinical practice, and this gap should be filled. And also, I would like to thank the organizers, because this is a high-level, serious forum where we see both clinicians and fundamental science specialists, and we need more fora like this. We need to work together. Yeah, but the problem, yeah, it is with us. Yeah, the virus will end at some point, but the problem of microbial resistance and safety, yeah, I believe this is a level of the national security, yes, and this problem will stay with us, and we'll have to deal with it many times. Thank you very much. Thank you. And now I would like to give the floor to Sergei Tsarenko, Deputy Head Doctor on Intensive Care in Moscow Hospital 52. And the same question, okay, this resistance.
Now, dear colleagues, friends, my friends, clinicians have said everything. Everything is bad, because we're treating with antibiotics, and my students would get very low grades. The 3 to 4 antibiotics we prescribe, expecting some sort of an additive effect, just a way to make sure that only 70% of patients die. This is a terrible antibacterial pizza. It's an atrocious situation. Plus, to what my friends have said, I would like to emphasize 2 more things. Apart from those obvious enemies, such as that mentioned, we should also mention the mycoses, and they're obviously there, and we can sometimes see the effects of that. And then one other thing, it is necessary to measure the concentration of the antibiotics that we administer, because very often we fantasize. So we begin to treat one patient. We prescribe a certain concentration of antibiotics. When we add kidney therapy, and we do not really know how much antibiotic there is in the system when the kidneys do not work. Then sepsis begins, and then, of course, the prescriptions become different. So we are treating with drugs that are not that effective. The schemes, the protocols are not that effective, and there's a very low understanding of the concentration of drugs in the system. So this is something to speak about. Yes, Carthage is to be destroyed, and the topic is to be raised. And one other thing to touch upon, organization. We should stop hiding the problems. We have sepsis in Russia. Yes, there is sepsis, and we shouldn't punish people for that. People should be able to inform about that, and the administrative decisions are to be changed. As when there are 8 patients in the ward, there are to be just 3. Then what kind of aseptics we're speaking here? That these are all very painful topics. Thank you very much, colleagues. As I said, this is a roundtable discussion, and of course I would like to give the floor to everyone who wants to comment. Yes, once again, from our viewpoint, the topic is most important. For different reasons, I should also say that the problem is a heavy problem. Indeed, the problem of the antimicrobial resistance will stay with us even when the pandemic leaves us. Now, let me say a few words. I would like to address all the colleagues who said a few words already. This is the old discussion in the field of molecular microbiology. When we're talking about the practical microbiologists, and Roman Sergeyevich, when they say the phenotypical manifestations of resistance are wider than what is known at the level of genetics. Nonetheless, today, there can be assays for very quick identification of resistance. The matter is that in the bed of the patient, particularly in the intensive care, it is possible to do it within just 1 hour. But there is one but. When we're talking to classical bacteriologists, they logically say that this is not a full coverage of the whole resistance, of all the phenomena that are associated with the bacteria in case there is classical testing carried out. Is it feasible to develop these approaches and these assays? And the community of those who come across multi-resistance, are these ready to test these systems in the clinics? Of course, not in order to substitute antibacterial diagnosis, but in order to supplement the set of tools that a clinic has. Let me try to answer. Now, let me start with the readiness. In spite of the past 2 years, we have all been working, and the COVID hospital in Kommunarka is the... yesterday, our scientific committee met, and it was stated there that at present our team is taking in 26 different projects, and 19 of them are already closed, and protocols, and 9 are dedicated to the new testing programs and the new protocols and new approaches. So not to consider the monocentric studies that we carry out, and we also publish the results. Well, of course, we're ready for that. Yes, we can detect quickly the mechanisms of resistance and not the agents. But sometimes we make mistakes, because with the mechanism of resistance, but without the activator, we can prescribe an antibiotic which is potentially can overcome this mechanism of resistance, but has insensitivity to that particular activator. So one does not preclude the other. So, of course, fast diagnostics is most important for heavy patients. We are familiar with the protocols that the patients with septic shock for survival need to get antimicrobial therapy within 1 hour after the onset of sepsis. So, of course, we must have assays for the proper selection of antibiotics for patients in bed. So these should be a personalized approach for the patients in critical state. Thank you. If you permit. I absolutely support what Denis has just said, because presenting the Sklissosovsky Institute, I can say that we're also involved in these studies, international serious studies. We have been active participants of that. But I have always been surprised with the following aspect. I'm talking about the gap between the applied studies and fundamental science. Why are we always getting oriented at the foreign assays rather than using our own? We do not have an enormous panel for express diagnostics. The drugs that we need in the clinic are dozens, maybe, in number. We understand what are the microorganisms that cause the infections that are accompanied with sepsis. There are just 6 to 7 of those, plus mycosis, of course. And then out of all the mechanisms of the resistance we have, of principal importance, not that numerous. And we need to examine the resistance. All this is very important for us to have a proper decision on the early therapy. And with surveys carried out through many years, I understand that in the regions, microbiological results are obtained by the medical doctors 4, 5, or sometimes 7 days after history, and the researchers. Well, after all, the pregnancy tests are sold everywhere. The express assays for COVID-19 are there. And I think we should start thinking about the context of antibiotics, because microbes are going to stay with us forever. Yes, thank you very much, Sergei Vasilievich. Your attitude is quite known, but still, could you say a few words? No. I'm ready to any result, but I do not share optimism from the point of view of the genes of resistance. Because unfortunately, we understand as a result, when we go along this pathway, that there is nothing to prescribe. We should understand better what we are treating. But if we understand that you are trying to treat and there is nothing for you to use for the purpose, then it's frustration. Classical diagnostics, as a result of monitoring, that's very important. We should understand what there is, what's happening in the intensive care units and specific clinics. And then the combination of antibacterial therapy drugs. And, of course, the assays must be express assays. Well, thank you. With your permission, in order to go back to the time for work, allow me to draw some conclusions. I would like to thank our colleagues, Denis Nikolaevich. We understand that you are extremely busy, but thank you very much for taking part in our discussion. Sergei Vasilievich is with us personally. I would like to thank for the roundtable discussion to Anna Popova. They said that I'm a classical microbiologist. I'm a classical pharmacologist, rather. We stand for everything that helps us to work with the patient and to treat the patient. If we work together in an active way on epidemiological surveillance, registration of sepsis, registration of different infections associated with medical aid, supplemented with phenotypical characteristics, with the technologies that we have in Russia, then everyone is going to be quite happy with that. Thank you. Thank you. We continue. Thank you. That was most interesting indeed and interesting.
Okay, now we are continuing our section, and the next is Mr. Bortnikov, and he's going to speak about circulating genetic characteristics of the coronavirus circulating in Russian Federation and Kyrgyz Republic. My presentation, please. Dear colleagues, the research of genetic characteristics of the coronavirus strain circulating in Russia began at the beginning of 2020. And in March, we got the first sequences of SARS-CoV-2 from patients in Russia. Then in June 2020, in the Vector Reference Center, we organized the molecular study. And this was till February 2021. And so the methodology was identified, and in Russia, a number of regions were identified under 9 research institutes of Rospotrebnadzor, and they were responsible for this total genome sequencing. And also there's a cloud service for uploading all the information. And in the Kyrgyz Republic, we began working with them in 2020, and this year we signed an agreement, and also there's an order signed by the government of the Russian Federation, and it is regulating sequencing of samples from the Republic of Kyrgyzia. Okay, so what can be said? So there's a certain level of laboratories, and it includes 112 Rospotrebnadzor laboratories. Now this number is larger. So they are responsible for diagnostics. And if there are some special categories, children up to 5 years, then recidival cases or some atypical cases, then they send it over to special institutions where full and fragmental sequencing is done, and then it is analyzed. And a report, there are weekly reports on the epidemiological situation in Russia. And these 2 upper levels, so this is actually this genetic monitoring that ensures identification and analysis of new genetic types of the coronavirus. Sequencing or analyzing of SARS-CoV-2, this is done in the reference center, Vector Institute of Rospotrebnadzor. So all samples come from the Laboratory of Molecular Research and Prevention of Diseases in Kyrgyz Republic. We have received about 160 samples, and the total sequencing... Right, sorry, I wasn't showing my slides. Okay, this is the scheme I was describing. Okay, so this is the genetic monitoring system, 3 levels. At the bottom, there are laboratories who do routine screening, and up there, genetic monitoring. The samples, as I said, from microbiological laboratories, and we have received over 160 samples, and sequencing is available for 140. what we have observed during these 2 years. In May 2020, what we studied, so those strains were similar to those were found in Sweden, Saudi Arabia, Russia, and Oman. But in April 2020, 8 samples, these were alpha, this is British, the British variant, and South African case as well. This is one of the previous ones that led to development of this current more aggressive strain. One was similar to the Indian strain, but not to the one that raises concerns. So those isolated types that were identified, so in what we find in Kyrgyz Republic. So these are different lines of families. In July 2021, we received only 23 samples and 23 types, and they are mainly from the Delta group, and this was prevalent in many countries of the world, and also 3 types which are similar to Delta. So we see that the Alpha, the British strain, is substituting with the Indian Delta strain. And Beta from South Africa was also circulating. So here we have noticed that certain parameters of genetic mutations, these isolated types of delta, so they can be clustered along 3 lines. So the sample volume is not very big, and so we can't speak about statistically significant information. But still, there are 3 different lines. Beginning from July this year, the cooperation with Kyrgyz colleagues has changed, and now we are planning to receive 100 samples every month. And in September, we noticed that all genetic types that circulate in the Republic of Kyrgyzia, so they are Delta 617-2. And at the same time, there are no Alpha or Beta strains because our colleagues described it that the Delta strain is not homogeneous and there are different variants of it. It is IY26 and 24. So again, this demonstrates the evolutionary process of world development of the coronavirus. In this slide, and you all are familiar with these pictures, we see the dynamics of spreading of this in Kyrgyzia and all over the world. We see that those mechanisms that cause substitution of one types with some other ones, they're typical for this republic as well. And now I'm going to describe what is happening in Russia. And it is not a secret, and it is clear that we observe approximately the same processes. As of the 3rd of December, the database had over 21,000 of full sequencing of SARS-CoV-2 genome. And what about Delta? In August, there was 96%, then in September, 95.8%, and October and November, over 98%, close to 99%. What is special about it? As we heard previously, in Russia, the most prevalent is Delta I1-2 and 617-2 and IY34 and so on. And in the Volgograd region in September and October 21, full genome sequencing, and this is the Indian strain, and this is actually 10% of all the sequences received from this region or studied in this region. So we cannot say that other strains have been removed. So those strains that caused a lot of concern, for example, the British Alpha strain in Moscow, St. Petersburg, Sakha Republic, Belgorod region, they were found there. Then South African, again, all these regions you see here. And gamma genetic variant, Brazilian, this is a rare strain. For the first time, it was identified in the Republic of Sakha. And in some regions, we see its circulation. And this is quite interesting. So deeper study of this variant in terms of epidemiology, maybe there are some national specifics, so this might give interesting results. Right, what strains are typical for the Delta variant? So B617.2, okay, it is well described. There's one Delta plus strain, and what is typical for it, the availability of mutations are shown here. It was first described for South African type, and it reduces the immune response. And this is IY1 and IY2 versions. And IY3 group at the bottom, it doesn't differ much from the original 612.2, but it is a different subline. And so there's some mutation which is determined not by S gene. And it gave a start to a new subline, which has mutations in ORF-B gene. All in all these changes, mutations that we study in the coronavirus, okay, they give certain advantages and pathogenetic advantages to the virus. This is the table with all the items, and it helps us understand what is happening with the virus. It shows that we may understand or observe only 1/3 of what we analyze. There we see nucleotide changes. So these mutations or changes, they are not so quick as for Ebola or flu viruses, but every change, every mutation in terms of the coronavirus, it might have a different and serious impact. And those mutations might lead to a super strain appearance. So, and Omicron, so this strain should be carefully studied, especially in view of the nucleotide changes. Okay, so these are the most prevalent mutations till October 2021. We also see that other wild variants, okay, they are changed and substituted by Delta variants. And this is in line with the world trends. And they are observed both in Russia and the Republic of Kyrgyzstan. And this scheme here, this graph, so we tried to analyze the main points in time when Delta strain became prevalent. So during this period, so this took about 2 months. And you see that a new Delta strain appeared, but a similar period was for D614G strain, and the same M142 mutation in Siberia, and then it circulated in other regions. And it took about the same time. So I suggest that all this massive metadata, okay, it may give us a chance to evaluate empirically new mutations and when they enter the population in order to assess the speed of spreading of strains that raise concerns that will be seen in the future. Okay, sorry, I kept forgetting, changing my slides. Okay, this is what I was speaking about. Okay, this is the time. Yeah, this 2-month period. Okay, thank you very much. Thank you for attention. Okay, now we are moving over to the next speaker, our colleagues from Belarus, Ms. Genesic.
So, genetic diversity and spread. Before I start my presentation, I would like to thank the organizing committee for the chance to take part in the forum and to share the experience of our work. So, incidents. We have over 60,000 cases. As for deaths, about 5,000, and there are about 10 million tests. I'm not going to speak about very general things, but on the 26th of November, Omicron was included into the strains of concern. But today we can say just one thing. The virus has a lot of substitutions, and some of them are quite well known. And I think that the potentially easier way into the sulfur virus is associated with that. And unfortunately, we do not have much evidence yet and much proof of the reality of all of that. We're carrying a follow-up of the individuals coming from abroad, patients who sick after the complete vaccination or immunosuppression, individuals with long-term infection and with getting the plasma, and we examine new samples to the type of outbreaks. We carry out the screening of the samples and systemically. And if there are individuals who work in the animal farms, we need to do something about the bodies of the animals. And we use several approaches for screening. First of all, when alpha appeared, we used one of the approaches. We developed a whole set of reagents, including primers. One group of those made it possible to reveal 6970 deletion and mutation 501 for the second group. No, second, the partial sequencing of S. And 3 complete genome sequencing on 2 platforms, Illumina and Oxford Nanopore. Now, the first cases were revealed on the 27th of December 2020. It was our center that was the only center in the country over several months carrying out the diagnostics for the SARS-CoV-2. And the analysis of the first samples demonstrated that they were grouped with the European samples, and the second one was related to the samples from Iran. And one of our first patients was from Italy, and we had a student from the second country. So it was EPID studies all demonstrated that. And March, April 2020, we had large diversity, a large number of genetic variants, and the imports were approved. They were from Russia, United States, Britain, Montenegro, some other European countries, and as Alexander said earlier, beginning with summer this year, Delta appeared, and all this became less interesting because really it's a dominating strain now. An interesting case. One of the patients revealed in July 2020, most probably he was a super spreader of the infection in Latvia, because practically all the registered data from SAIT is associated exactly with that patient and the variant typical of him, and that particular variant went to other countries as well. Now, the Nigerian type of virus, family couple, they came from a holiday in the Emirates, but this virus did not go any further. And so, but we had a fact, and as for the variants that concern us in our country, there were three, alpha, beta, and gamma. And the dynamics of spread of alpha in Minsk showed that starting with February, there was an increase, and February, March, April, 80% of cases were alpha associated, and starting with May, we can see that all the alpha went down almost to zero, and since August we have not had it. As for Delta, in May for the first time, and now that's what the substitution occurred like that. Delta spreads very quickly. Our colleagues were saying that in Russia, the substitution of alpha by Delta occurred within 1 month. In some countries, there was probably a similar situation. But if we take our country, here is the analysis for June, July, August, September. And in all the regions of our country, starting with August, there was just Delta manifested and no other strain at all. If we look at the genetic diversity within the subline, the picture is somewhat different from the one in Russia. At present, almost more than half of cases are related to the initial variant of Delta. So it's AY79, AY5, AY33, 39, and only one patient had 122, which is so widely spread in Russia. Maybe with time the situation changes, but at the moment that's what we have. Now, here is this curve of incidence, and you can see the alpha, delta, and you can see that it coincides with the increase overall number of cases. We're talking about Omicron, but at the present we do not know what the epidemic potential is, and we do not know whether it is going to substitute the existing options or not. And Omicron. Now, we're going to share our experience.
Okay, and these are different types. Yeah, so this is characteristic for Omicron. And if some country doesn't have the basis for such large-scale research, this preliminary screening can be done using this deletion. And I would like to thank all the specialists in our country who are collecting this, and I'd like to thank Ms. Popova, Mr. Kimkin, So we cooperate in the area of this full sequencing. Thank you very much. Thank you. Since you followed the time frame very carefully, I will ask you a question. You mentioned that in Belarus there are no such opportunities for this fully fledged sequencing. At the same time, you have certain perceptions how different mutations in Delta strain and other strains that were circulating previously, how they can contribute to the qualities of the virus. Are there any experiments, maybe, or calculations, or modeling that can evaluate so-called fitness mutations? And it's not only about structural proteins, but proteins which are non-structural. I'm not speaking about ASP, but only about structural proteins. Are there any factors that can forecast the replicative or toxic features of the virus due to all the data you have demonstrated? Well, we can do sequencing, maybe not full genome. So we didn't have great opportunities, but currently we are developing and we use this method. As for your second question, we haven't been doing such focused research works, and we work on CL3, and we study neutralization, viral neutralization with antibodies. There's this study as well, and there are two findings. So we have received sequences. It is when a cat transferred or a cat contracted a virus, and a mink. So it is cross-species transition. So we have all this. And as for fitness mutations, we didn't study it yet. Okay, colleagues, are there any other questions? Right, now let's move on to the next. It is Mr. Dudkov, and who is going to give a talk on a similar topic, but he will focus on the northwest federal region of Russia. Thank you very much, Vadim Markovich, for this opportunity to speak. My report will be in line of similar presentations, so I decided not to focus on genetic specificity of different strains. And I'm not going to cover this in detail because you have listened to it and heard about it several times. We are now in St. Petersburg, and St. Petersburg is the center of northwest federal region of Russia, and we are a federal we are a branch of the federal institution, and we are responsible for this region, and we do all this genetic and virological monitoring. And I wanted to identify a number of specific features. So we have 11 entities or sub-regions, but they're all different in terms of their contribution into the epidemiological process. Of course, St. Petersburg, as the second largest city in the country, its contribution is the biggest and largest in this northwest. We also have Yamal-Nenetsk region. Their contribution is close to zero, and although there are some cases registered there, and there are some regions which are similar to St. Petersburg in terms of value. If we talk about the incidence, it is similar to St. Petersburg, and in some regions it is even higher. So this is an interesting observation because the density of population is much lower, but at the same time, the intensity of transfer is very high. Speaking about the severity of cases, and these slides I'm showing to you, this is the current situation. Okay, and this is what has been happening during this week. And we see here that the light cases are prevailing, but in Novgorod and St. Petersburg, these are two exceptions. So there are fewer severe forms, and without symptoms, again, not very many are identified. But this is probably a problem of identification, diagnosing, and you will see some proofs for that a bit later. In terms of the age group, again, it is about the same in all regions. These are people at employable age. At the same time, there's a growth of incidence among children. Taking all this into account, we decided to summarize all epidemiological studies and environmental monitoring data, and we decided to have this overall picture. So this slide, it's not, to show how many different genome types were identified, but no, it is just to stress that we used a Rospotrebnadzor database, not the open data, but those reports that the central administration, central management of Rospotrebnadzor receives. So looking at morbidity and its dynamics throughout the year, it is similar to what we observe in the country. Okay, there are like waves of incidents, and as I said previously, St. Petersburg is the second largest city in the country. In terms of the age groups, all in all, people of active age are infected, and recently children were added to this. Right, in terms of genetic diversity, yes, this is interesting. This diagram demonstrates the picture since April, because this is when the 56th order was issued for planned genetic monitoring, and it is when there were unified rules for this process. Before that, it was done sporadically. So, for example, something we received into our diagnostic laboratory, or when there were some special cases. And in April, we had quite a heterogeneous image. Okay, lots of different strains, beta, alpha, and northwest strain that was first identified in January. But again, I'm not boasting of it. Yeah, but this sample, it was dated January, and although other colleagues in other countries as well, okay, they published their sequences, but there was no single sample with the sequencing that was collected before January. So it was identified in this region the first time, but probably there was some circulation of it before that. In May, we heard it several times already, we got Delta strain at the beginning of May. It appeared here. Okay, the numbers were growing. And then in June, it became prevalent. and then all other strains were substituted in August, so that this strain was dominant without any exceptions. Have a look at this slide. Have a look. This is mortality, and this changed as well. If you look at this, see that in May and June, okay, it was very high, but then in July it fell down and it was getting lower and lower, and now, okay, it is half of what we had previously. Let's keep it in mind.
Something to be remembered. The same is true for the spread of cases by the severity. No, by present there is a reduction of the number of severe cases. This began in April and May. Let's remember that. Now, let me touch upon an interesting aspect associated with the dynamics of the northwestern strain, which, as I have said, was revealed by our staff. And it's obvious that it's better to have a proper sampling for the analysis. Sequencing doesn't help much, but we developed a method that makes it possible to work in real time with PCR tests, so we could check many samples and to see the percentage difference and changes, and as a result, the spread of the strain. Why did we pay attention to this particular strain? It had some specific features. There was long deletion and a long insertion, and from the point of view of phylogeny, we could not associate it with other strains in our territory, which were all with the strains sequenced abroad. But nonetheless, there were two sequences revealed. One was from Pskov region, the other one from St. Petersburg. And these people were not connected at all to each other. And this was checked. And that means that the strain circulated. In January, we found it. But by that time, it had been in our area for a while. But for how long, we do not know, because it's impossible to sequence samples for the past. And so the phylogeny is based on the strain classification. We can see it's a separate branch, and as for the number of manifestations of the strain in Northwest increased in Pskov, Moskovsk, Moscow region, Saratov region, as well, as far as I know. Maybe not. Well, you can, and you should correct me. The biological properties should have changed, and we're terribly afraid to see a geno variation with the mutation. No, there is mutation. Something might happen. We believe here is a 3D image, and you can see the substitutions there. Now the slides are not being demonstrated. Yeah, I forgot to change the slides. Now you can see the locations in the protein structure that changed, and so the biological properties of this object should be somewhat different in theory. But if we look at the dynamics which were assessed with the representative samples, we can see that there was a growth. The share of the northwestern strain in St. Petersburg increased to 5%. And here we even wondered when it would overcome all the others. But then Delta strain appeared, and the Delta strain absolutely pushed it out. So the strain here in Northwest tried to resist, and the but the last manifestations were at the end of June. In sequencing, we're unable to see it, but nonetheless, we're looking at many samples, and we were able to find some separate cases. So I would like to mention that with sequencing, it is hardly possible to say that the biological properties are strongly different and changed. We shouldn't be afraid of that, indeed. And what is peculiar in this story? When we were unable to manifest that strain, then WHO found it suddenly. That speaks a lot for the attention paid by the WHO to different strains. The WHO declared that it was this strain was deserving a matter of attention, but then after a month it was removed from there because it had disappeared. But by that time, we had known much about it. Then there was another case of interest, a co-infection. As everyone says, the co-infection, what is co-infection? Theoretically, co-infection is possible. But I wonder if anyone examined it in a practical sense. So we decided to have an experiment, and we used for the purpose a custom panel in order to check the respiratory pathogens and COVID, a lot of pathogens, including COVID. And we selected representative samples from people with respiratory virus infections, not the COVID, but with the respiratory infection. And from outpatient clinics, and so they were sent to us for etiological diagnosis. At the initial stage, we carried out the PCR tests. In 45% of cases, COVID was revealed, and then after that, we used the panel and the chess method, and in 86% of cases, COVID was manifested, yes, and was found. So it seems that half of those cases of COVID that taking place actually in reality are not visible to us. The NGS method is much more sensitive, and so much more sensitive than PCR. In 6% of cases, we had co-infection. Or I would rather say that there were certain symptoms of co-infection. In one sample, for example, we were able to find two types of nucleic acids. So that's what it looked like. So we were able to find seasonal human coronaviruses. We were able to find virus of flu A, paraflu of type 2, and some other infections. So about 46% of people with COVID have plus to that an extra co-infection. So it appears that the simultaneous infection with two different viruses is quite possible. Theoretically, we understand this, of course, but instrumentally, we were able to find the proof. Now, on the next slide. Now, could you? Yes, the final two slides, and that will be the end of it. This slide is quite provocative. It just shows that there is a big lie in statistics. No, that's about the fact that among the people who had the disease, the number of the vaccinated is larger, but there is a much larger number of vaccinated now. And among these people, some fall ill, and they are of particular interest for us. But these people go to a specific study and research, and that's why for us it seems that the number of the vaccinated people who catch the disease increases. this is not really so. And the severity of cases and the death rate. Here you can see one of the reasons, along with the growth of the number of vaccinations. Although Delta virus is more pathogenic, more contagious, the severity does not grow, and the lethality does not grow, although the number of cases increases. So if we carry out the vaccination program in a systemic way, then Delta and Omicron are not going to be that terrible for us. Allow me to introduce the final presentation for the session by Sergei Balakhonov, who will speak about the dynamics of the genetic structures and phylogenetic reconstruction of SARS-CoV-2 circulating in the entities of Siberian and Far East federal districts. I know this is the final presentation, and after the time allocated, so I'll try to be quite brief and tell you the story of what concerns the genetic structure and the changes of the genetic structure in the process of the spread of the COVID in the areas we are responsible for. First of all, a couple of words to begin with, although many have mentioned the pathogen, contrary to the earlier impression, is quickly changing and quickly modifying, and this global evolution and spread lead to the consolidation of new mutations in the key sections of genome of the virus and to the emergence of the new genetic variants with certain properties that increase its epidemiological significance. As for Omicron, and B.1.511. A lot has been said about this, but this emphasizes once again how important the monitoring is of the circulating strains of SARS-CoV-2 for the proper assessment of the epidemic processes and for the description of the genomic changes that lead to the increase of this pathogenic character. Vasily Gennadievich spoke about that, and I'm not going to repeat what has been said. But there is a unified approach here, and our institute, as a regional center for 8 entities mentioned here in the slide, carries out the genomic sequences with Illumina-Seq and whenever, and the fragment analysis on S protein as unified Arctic protocol. From January to November this year, we examined over 900 samples and sequenced 1,700 isolates, and alpha, beta, and delta were revealed. And in the structure of these variants, delta dominates. Let me go back to the previous slide, which is more specific about that. And even at the initial stages of research in December last year, in 100% of cases, we were able to find the initial genotype. Then alpha was identified in March. In certain samples of June, eta and delta for the first time at the end of April, and by July it was identified in 90% of samples, and it pushed out all the other variants. So the same is true for all the other areas in our district, with certain variations in time. I should also say the emergence of the variant Delta, and this coincided with the increase of the number of registered cases of COVID-19 in the areas, respective areas. The blue line shows the dynamics of the new cases, and the analysis of the genetic lines, according to the Pangolin program demonstrates that subline 1 to 2 dominates, and the widespread of this genetic line was typical, as it has been said, typical of the Russian Federation. 45% of data in the database of sequenced genomes is associated with this line. So there is nothing unusual in that, in spite of the fact that the areas we're talking about are isolated and are really remote areas. In order to understand the logic of spread in these areas, we carried out the analysis using the FST criteria. The higher is the FST, the higher is the isolation between the neighboring territories, and the results of the analysis are presented in the shape of the graph, where you can see the wider is the line, the highest the FST. The maximal FST is typical for the Altai district and Buryatia, and the most isolated of those is Altai. Irkutsk region, Buryatia, Zabaikalia, we can find a free circulation of the virus. But on the whole, the importance of FST is not that great. The level of FST is not that high, from 0 to 1. And the virus can accumulate quite a lot of nucleotide substitutes for better calculation of the criteria. And in this sense, the analysis of the genetic diversity expressed through HD is important. And it's the possibility that two sequences might differ by one nucleotide. So there will be two different haplotypes. So SARS is less variable in Altai region. And so there is some statistics presented here, but this logic was found nonetheless. Then, at a later point, we carried out the reconstruction of the dynamics of the genetic diversity of the virus in all the 8 districts. The blue area reflects the trust interval of genetic diversity. And the dark line is very important here. It can see that the curve of active cases repeats the dynamics of follows the curve for the genetic diversity. And then at the very end, we can see that it coincides with the minimum number of infected at the analyzed areas. At the beginning of June, we register the growth of the incidence, and along with that, the genetic diversity also increases. Most probably, it is associated with the fact that Delta strain appeared. The earliest manifestation in Buryatia was registered on the 22nd of April, and then on the 28th of April in Zabaikalia, 30th Altai, then Krasnoyarsky Krai, and so on and so forth. And the latest is Khakassia on the 27th of May. And so these genome data make it possible for us to conclude that Delta needed just 1 month to spread and consolidate its potential in these areas. Here you can see the data for the analysis of the variability of proteins in the isolates of SARS-CoV-2. Now, the left graph over here shows that the RFFID protein is most variable. And if we calculate everything and use the relative numbers, then RF7 and B and A should be mentioned here. As for the significance of the proteins S and nucleocapsides, now we carried out the deep analysis of the dynamics of their changeability. And as a result, we were able to see that from December 2020 up to November 2021, in the absolute expression, the proteins S and N accumulated less substitutions than AB polyproteins, and in the relative numbers, it was became typical for nucleocapsides. For the analysis of the dynamics of the amino acid substitutions in S protein, several mutations were selected typical of each of the VOC variants. And of interest here is the registration of the typical VOC variants before the emergence, actually, in the areas studied. By different lines, we can notice here that we can see that the substitution of PH81 are typical for Delta. About 20% here was registered even before Delta appeared as a strain. And the particular diversity of this substitution in S protein is noticed in April and May. 12 and 11 substitutions when all the three variants appeared in the areas. And then as a result of the total domination of Delta, the significance of the substitutions went down. In September and October, the sequencing of several samples associated with Delta containing the substitution of Q line went, and isolates were found in Krasnoyarsky Krai, and one in Irkutsk, and in November the N501G. This line is there, but it manifests itself in the structure of nucleocapside protein. There were 4 amino acid substitutions, 3 of which were found in 98% of the isolate Delta, and 72% when we carried out the screening of the genomes presented. Here we can see some phylogenetic trees reconstructed in compliance with the program, and we can see here that Delta prevails here. The territorial clustering is not typical of that picture, which correlates with the earlier data. And still, we can see 2 collides here, owned by the isolates from Krasnoyarsk and Irkutsk in the lower part of the tree. And what else should we comment on? The comparative analysis of the genomes and clusters shows that the amino acid substitutions are typical for Irkutsk region and RF7A. And the frequency here is 100% and 99% respectively. But in Krasnoyarsky cluster, these substitutions they are not present. In half of the sequences from this cluster, there is a mutation H358. Now, in the Irkutsk cluster, there is a subcluster for all isolates with AV570S, and then possibly there is a reorientation of the receptor binding domain to the open conformation, open for the binding with the AS2 receptor. According to the BRCA4 portal, the variant of the activator with this type of substitution occurs in 1.5% of isolates of AY122. And it is necessary to mention here that SARS-2 with this mutation is identified only in the toxic material from the people who died of COVID in the Irkutsk region. And now, here is what concerns the substitution in the protein structure. And now, conclusions. We need to say that in the dynamics of the pandemic, in these isolated regions of the Russian Federation, in Siberia and the Far East, we were able to find the trend typical of the Russian Federation, already described today by Vasily Gennadievich. And the dominating strain changes, and there is also a genetic diversity of the circulating Delta strain. Identification of phyletic clusters, SARS-2 requires additional examination in order to understand the spread and the potential impact of these markers on the epidemic process. That's it. Thank you. I'm sorry. With the deficit of time, it was difficult. If there are any questions, I will be able to clarify things. Now, colleagues, we have come to the end of the session. Unfortunately, we're outside the time allocated, but it was quite interesting. Just a couple of minutes to conclude. We won't have a discussion. Our session demonstrated that, and the plenary presentation of academician Kinkin, that we have a lot of information. But I'm expressing the opinion of those who are listening to it. But the depth of this information, the processing of this information is insufficient. We have accumulated, with the use of different molecular methods, a lot of raw material concerning the viruses, the replacement of the strains of some strains by others, and the situation in the clinical situations in combination with different infections, co-virus infections, bacterial and mucosal. But all this requires detailed attention and detailed discussion, analysis and extraction of the new data. Well, I might be quite provocative saying that for a while, without lessening our attention to the molecular observations, we should think of the new knowledge that we might need for clinical doctors and for administrators to cope, because sequencing and bioanalysis, that's just a mechanism in order to get a new picture. And I think that within the framework of our conference and the roundtable discussions, within the framework of the period we will have after the conference, it's quite important to carry out this work, because it's most valuable information which is just there without being used. We have specialists on coronavirus who can help with the forecasting of dynamics and with the the assessment of the value of different substitutions. And one final thing to say, apart from the structural proteins we're all examining, believing that they are targets for the vaccine antibodies and for the immune system. But there is a much more threatening story not mentioned, the mutation processes that concern the host proteins and episomal. And so, since the usual methods of typing built on PCR and PCR techniques are giving way to the polygenome sequencing, we should be very attentive to these issues. Much more attentive we should be now, because mutation in episomal will make the situation with the virus mutation very different. It is well known that this virus has a good apparatus for the support of the genomic homeostasis, but as soon as selection occurs of mutations that allow it to make mistakes and generate different variants capable of replication, the evolution will be much more rapid. We break for lunch now. And now we would like to meet at 3:30 here.